So today I'm going to talk about novel and old perspective on the structure function dynamics and human and non-human primates and the diverse states of consciousness. And I will try to be more precise on what do I mean by structure function and even consciousness. What do I mean more precisely? So let me start with
I think you need to click on got it.
Okay. So, in this talk, I'm going to present three different perspectives on this problem. And again, the perspective on this structure function interdependent modulated by the different states of consciousness. So the three phases that I'm going to talk about first, it's a topic that you know here in this lab is functional dynamics. The second one is something that you are probably also aware of because it's a collaboration with Mathieu Gilson. It's multivariate Ornstein-Uhlenbeck processes fitted from fMRI data in humans. And the third one, it's connectome harmonics. So as I say, I will try to be more precise and what do I mean by structure, what do I mean by function, and what do I mean by consciousness. So let me start with the easiest one, consciousness. The more complicated one, I'm not going even to attempt, try to define what it is, but at least try to provide a framework in which how we, computational neuroscientists in modern times, were approaching this problem. So first question that I like a lot is what kind of scientists study consciousness? And I would like to start with this historical approach that I like very much from this phrase of Stanislas Dehaene. For many years, no serious researcher will touch the problem. Speculating about consciousness was a tolerated hobby for the aging scientists. And just if you already got your Nobel Prize, you was kind of allowed to speculate about consciousness without being considered a charlatan. But now things change because we can now measure brain activity more precisely. And we can start to ask some basic scientific questions that can be answered from the scientific perspective. So this is something which is new and has changed in the last years, thanks to technology and thanks to science in general. So why study consciousness? This is an important problem, a societal problem, because we have loads of people with accidents that goes into the states of coma, and we don't know how to get them out. We don't even know what's going on. There are different ways in which you can enter the states of coma, apoxia, different brain lesions, among other causes. We have a more aging society getting problems of dementia, and we don't really know exactly what's going on in the brain. But we have, again, a society that is getting older and older, and we don't really know what are the different categories of consciousness of people under different problems of neurodegeneration. We have also a society which is more stressed and have all different problems of mood, and when we don't really know how to help them, and we know that there are different traditions that have different techniques, but we don't really know what's going on in the brain of these people. And as we don't understand really well, so far the science of meditation in general is not really well established. We have one of the most wonderful drugs and probably one of the most important discoveries of the humankind is the discovery of general anesthesia, huge development in medicine in general. The modern notion of hospital is not even conceivable without general anesthesia. All the development of cardiology, it's not even thinkable how the society will be today without general anesthesia. And there are different drugs of general anesthesia, and we don't really know how they work in the brain. And there are a lot of surgeries or surgical procedures that are not done in people with different conditions or aged, or because we don't really know and understand what's going on in the brain. Different conditions like catatonia and families devastated by this condition. And again, we don't really know how to treat the patients in this condition. Society, again, more stress, having problems of sleep and all the problem that comes with problems of sleep. We were talking about memory consolidation, among other problems. And again, these are different states of consciousness in which humans go every day and we don't really understand. And again, we have different drugs that can alter the state of mind in general and the state of consciousness that can be extremely helpful in that are resistant to classical treatments, psychotherapy, but also to the classical drugs that are used to face the problem of deep depression, and they are treatment resistant. And today we have more and more knowledge about psychedelic drugs, but still we don't really understand what's going on in the brain in detail. So that's why I think it's important to try to study consciousness from the scientific perspective, using the technology and that we have at hand in modern times. And how to study consciousness? Well, consciousness has been studied from a long time, from millennia, actually, from the perspective of the religion and the shamanic traditions and spiritual development people. But also lately in psychiatry, using different approaches. But science, hard rock sciences, it's more new. And even neuroscience, and that's important for the historical perspective, but probably the first one that tried to locate consciousness in the brain from a scientific perspective was René Descartes in the 17th century. He was doing neuroanatomy and trying to place the site of the brain in which consciousness is located. He concluded that the consciousness must be located in the pineal gland. Of course, now we know that it's not the case, but the effort and probably the first figure that tried to locate a place in the brain where consciousness is located is René Descartes. Of course, now we have more technology and different approaches that allow us to better approach this question. So, of course, this is working. So, how can we investigate scientifically this organ in different states of consciousness? Well, the question is, it's complicated just at the level of the scale. So, it's consciousness located at the molecular level. There are even Nobel Prizes trying to postulate theories related to molecular processes responsible for consciousness. It's at the cellular level. We don't really know. It's at the level of the circuits of the different neurons. It's at the level of the brain regions, probably as Descartes was postulating, or it's at the level of the whole brain. And of course, this question cannot be answered clearly, but studying the whole brain is probably the less ambitious way to approach the problem, because we are not obliged to locate consciousness in a specific place, but trying to find neural correlates that are global at the level of the brain that show the evidence of the presence or absence of consciousness. So the first research method that I'm going to talk about is fMRI, functional magnetic resonance imaging. Just to mention that to build this machine, you need 7 Nobel Prizes. So it's wonderful achievement, scientific and intellectual and engineering achievement. It's not just that the ideas need to be set up, also the engineering behind this machine. It's really incredible. And probably for your surprises, we're not measuring directly brain activity, not activity of neurons. We indirectly measure blood oxygenation in what we call the bold signal, which is this response of the brain in terms of blood oxygenation. As you know, more energy requirements are needed when the brain is involved in a task. So when the brain is involved in a task, you need more oxygenated blood, and this can be measured thanks to what we call the bold signal. So the MRI scanner is able to measure changes in magnetic field caused by difference in magnetic susceptibility of the oxygenation of the blood. So at the end of the process, you have a signal that it's in every voxel, so a volume of the brain that it's in time. So if the task is visual, you will probably have the visual cortex showing more contrast in the task. If the task is hearing, you may have more activity in the auditory cortex, and so on and so forth. We are interested in consciousness mainly in resting state. So when the patients are asked to do nothing in a sense, so they're not involved in any active task. But at the end of the day, what we end up working with is these signals that are like following each of the voxels in time, and we have parcellations of the brain. So we group different voxels together following anatomical maps or functional maps. And at the end of the day, what we work with is these bold signals that are the changes in time of the level of oxygenation of each voxel or groups of voxels. So more or less, I introduce what do I mean by the different states of consciousness that we work with. Now I'm going to try to focus on what do I mean by function. And by function, we mean pairwise correlations. So usually what a bold signal session or sequence of scans can look like this. These are 234 regions, bold signal fluctuations in time. And what we have here is functional connectivity. So pairwise correlations in the whole time of the experiment. So what we have here in this matrix in the 2060, let's say, is the 20th signals that correspond to a brain area correlated with the 60, that is another one, and you compute the Pearson correlation between these two regions, and you assign that would be a number that you can assign a color. And at the end of the day, when you have all your regions correlated with all of them, so pairwise correlations, you have this object that we call functional connectivity. And it's very informative, as we will see. But the problem is that if instead of considering the whole experiment, you go and consider time windows, what you see that is that the brain in the different states of consciousness, it's a very dynamic object. So it's probably not a good idea to take the whole window and consider one set of pairwise correlations. It's probably a better idea to consider sequences, time windows, and see how the correlation change in time. And this is also a common approach that is called functional connectivity dynamics. That is, again, instead of considering the whole window, you consider windows of small size. Of course, here we have the trade-off is that if the window is too short, we are not really considering correlations. And if the window is too large, we are considering less windows because you don't have infinite amount of time, especially in both signals that we have not long enough recordings, and this temporal resolution is very poor, but we do what we can. And this is the problem. okay? And if you consider the correlation between the correlations in time, you end up having this object here that is time windows, that is the functional connectivity dynamics. And it's important because we see that if we consider temporal windows, we see these patterns that appear in time. So this is the brain of a macaque in the awake state. And we see that we have seen this pattern before. It appears previously in time. So here, in principle, we don't have like an infinite amount of patterns. There is probably a bunch of them. So a good idea would be then to try to cluster them. And we will talk about that in a minute. So what do I mean by structure? And this is the last component of the three things that I say that I'm going to try to clarify for you. Structure in this talk will be the anatomical connectivity measured by GTI. So what it is this more precisely? It's a technique of MRI in which you can track the diffusion of water molecules, the anisotropy of the diffusion of the water molecules, so the preferred direction of diffusion, and the preferred direction of diffusion, usually the white matter tracks in the human brain or in the macaque brain. So at the end of the day, we may end up having this object over here that we call this is white matter tractography. But at the end of the day, what we end up having and working with is with a parcellation of this object. We end up working with what we call the connectome or the matrix of anatomical connectivity. This matrix is usually counting how many fibers enter, or start in one direction, in one parcellation, and end in other parcellation. In humans, you cannot distinguish actually what means start and finish. You just have a symmetric matrix of white matter connectivity. And of course, this matrix can be normalized, and what does it mean exactly, this number? It can be many different things, but in principle, it's a kind of normalization of the number of white matter fibers that are detected by this technique and connect to different brain regions, where brain regions really means depending on the parcellation that we are working with. So this is what I mean by structure. So now we can try to answer this question. How is the relationship between brain structure and function modulated by the different states of consciousness in macaques and humans? And this is the first thing that I'm going to talk about, functional dynamics, and how this can be revealed using this technique in different data sets. So the first data set that I'm going to talk about is a macaque in different under different type of general anesthesia. This is a wonderful work, experimental work done at the Neurospin by Bashir Haraya and Lynn Urik. That is 20 different sessions, more or less, of macaques in the awake state under light propofol, light sevoflurane, different drugs, ketamine in the general anesthetic dose, deep propofol, and also deep sevoflurane. So we have, these drugs are really different. The way in which they act in the brain is different, but they all lead to this state of general anesthesia. So complete unresponsive of the macaques, responsiveness. So the first approach is the static. So the static picture, what is the structure function correlation modulated by the different states of consciousness in the static picture is, it's simple. You only need to correlate this matrix. So you transform this matrix in a vector. You transform this connectome in a vector. And you just compute the correlation between these two vectors, and this will be a number. And as you have more or less 20 different sessions of awake macaques, you can compute these numbers and put them here in a distribution. So you can do exactly the same procedure for ketamine. And look at the differences. What does it mean? It means that the correlations, pairwise correlations, that you find in the states of general anesthesia, pairwise correlations, are more and more similar to the anatomy of the brain, white matter fibers. So something which is completely anatomical start to resemble the function. What does it mean? That it means that the correlations that you find in the states of general anesthesia are heavily influenced by the anatomy and not that much in the state of wakefulness. So the correlations that you find in states of unconsciousness are heavily driven by the structure. And it's not just a coincidence of ketamine. In low propofol, this is what happened. In deep propofol, this is what happens. And so on. It's the same. So there are significant differences, statistical differences, what you find in the awake and the rest of the anesthetic case. And again, this is a static picture that I already criticized because, as we see, there is a lot of fluctuations in this object over here if we take different time windows. So how we can try to assess the same type of question, but now modulated by the different states of consciousness, and also considering the fact that inside the static picture, there is a lot of dynamics. One of the classical approach is doing the following. You put all the scans that you have in the awake state. You concatenate them with all the scans that you have in the low propofol scenario with the deep propofol scenario, and you have one standardized. You need to set score and apply other features, but at the end of the day, you have a huge database with all the scans in the awake state, all the scans in the low propofol and the deep propofol. And then what you do a sliding window approach to this object over here. So instead of having a few functional connectivities in time, now we will have a lot because we'll have all the patients in all the conditions. Okay? What happens if you do that and then try to apply a clustering algorithm to these objects over here? You will end up with centroids. Okay, that are the centers of this K-means algorithm. And instead of having what we usually call functional connectivity, you will have these objects over here that, by the way, they look a lot very similar to the functional connectivities that you find in the different states, but let's don't focus about that. And the other thing that you can do is to rank them from the less similar to the structural connectivity. This is the connectum of the macaque. This is the less similar centroid of the K-mean, and this is the most similar centroid to the K-mean. You can rank them again. You transform this in a vector, compute pairwise correlations, and rank them from left to right. What another output of the K-means is the sequences of the state visited by your time windows, okay? My first time window was more similar to state number 4. My second one was similar to state number 5, and you have this sequence. And then what you can do is in all the first part of your experiment belong to the awake state. So you can make an histogram of the state visited by the first part of your experiment. And then the second one moderate sedation, and deep sedation. And here there are two things that are very interesting that happens. First of all, that in the states of anesthesia, you have a reduced repertoire of the state that you visit. So you lose these two states. Okay? Second thing that is very interesting, look at the states that are most visits. It's the number 7. But the number 7 is the closest to the connectome. So in the states of general anesthesia, the most recurrent pattern that you visit, as revealed by the K-means algorithm, is the state that resembles more to the connectome. Okay, so we rediscover again using a different technique. The same fact is that the function, it's driven by a structure, or at least that the correlations that you find in the anesthetic state are heavily influenced by the anatomy. Second thing, and this is a very solid result, is that the fact that you can rank here the states by the similarity with the structural connectivity, and this will give you a similarity score. And then you can put this histogram here in this 2-axis probability of visits of this histogram. But here in the x-axis, you can have the structure-function correlation or the similarity scores between structure, and then try to do a linear. The result here is that the more you go to the unconscious state, the more steep, the more pronounced is your slope. And we will see this result again and again. Okay. Second, very beautiful experiment that we are working with is called deep brain stimulation of the central thalamus. Here is the deep brain stimulation in the macaque. This electrode that goes in the brain have different contacts. This one goes to central thalamus, and this one to ventrolateral thalamus. This is more or less the experiment. The animal is awake. The animal is in the state of general anesthesia, so completely unconscious, unresponsive, and everything. And then electricity is turned on in the central thalamus at 3 volts. Electricity is turned on at 5 volts in the central thalamus. Under general anesthesia, continuous infusion of propofol. You change the leads now, it's number 3. Ventrolateral thalamus, 3 volts. Ventrolateral, 5 volts. More closely, these are 2 different animals. This is the central thalamus in the 2 different animals, and this is ventrolateral thalamus. Just for those who don't know, central thalamus projects like crazy to the rest of the cortex, and not so much ventrolateral thalamus. So stimulated here, the activity will propagate to the rest of the cortex, not that much ventrolateral thalamus. What happens at the behavioral level when the animal is awake, the arousal score, when we measure the exploration of the surrounding world, spontaneous movement, shaking, prodding, toe pinch, it will react, eye-opening, corneal reflex, all the scores are high. What happens in general anesthesia? You lose everything. You are in the general anesthesia state, completely unconscious. What happens if I turn on in the central thalamus the stimulus at 3 volts? I start to gain some of the behavioral responsiveness. What happens in general anesthesia? Again, when there is electricity in the central thalamus at 5 volts, boom, the animal is awake. This is really impressive. What happens if you miss the target in a way? So instead of targeting central thalamus, you target ventrolateral thalamus? No much happens. So it's not just put electricity like crazy in the brain. It's targeting the right place with the right voltage. So this is a nice result of in the lab of Monsieur Haraya, Stanislas Dehaene, and colleagues. That's is advanced published last year. So we look at this data set from the perspective of the static picture, and again, we found what we described before. If you look at the structure function correlation from the static perspective, again, take all the functional connectivities, all your scans in the awake state on the 3 volts, 5 volts, central thalamus and control, and you find that in the awake state, your correlations are pretty weak if you compare them with the off state. Off meaning general anesthesia and no electricity at all, so general anesthesia. On 3 volts, on 5 volts, 3 volts control, on 5 volts central thalamus. It resembles awake, as it resembles in the behavioral arousal scores. So this is a kind of solid neural correlates of the state of consciousness as we see. But it's not that. What happens if we start to look at this problem from a dynamical perspective? Look how the functional connectivity dynamics looks in the awake state. Look how it looks in the off state. General anesthesia, much poor dynamics here, just in diagonal. In on 3, we start to see some activity. Look what happened on 5 volts in the central thalamus. Again, start to see again different correlates of the state of consciousness. What happened in 3 volts control? So ventrolateral, nothing, really similar to off. If we increase the level, but in the wrong place, wrong control, nothing happens. So again, in this article, they perform different experiments using different paradigms, like a local globe that I can explain probably at the end. If you have a question, frontoparietal singular cortex connectivity that is always another correlate, but also K-means clustering using the sliding window approach. What happens here is, again, as I said before, awake is characterized by a slope that is much less than in the unconscious state. This is general anesthesia. This is when the lids are off. General anesthesia. What happens when I stimulate central thalamus with high density? I come back more or less to this level. What happens if I go from anesthesia, from the awake to anesthesia, and then I stimulate the wrong place, ventrolateral? Well, I don't come back. So again, this is a robust signature, as we have seen before, by the different type of anesthetics, ketamine, sevoflurane, and propofol. And we see here again, even when we apply central thalamus, we recover the flatness of the relation structure function that we lose under general anesthesia. We apply the same, but using the same approach, using sliding windows. But the problem is, again, one of the critiques that I mentioned before is how to choose exactly the length of the time windows, given the fact that we have this problem of trade-off between the length and the number of. And there is another approach that is very nice, that is based on the Hilbert transform of the Bolt signals. You can think about these Bolt signals as the shadow or the projection in the real axis of a more general signal that is called the analytical continuation that lives in the complex domain. And in this complex domain, this quantity, that is the Bolt signal, can be decomposed as an amplitude and the phase, cosinus of the phase. So we can recover this shadow of the real axis as the multiplication of the amplitude and the cosinus of the phase. Okay, so instead of having one connectivity matrix per time windows, what we will have now is, as we have one phase per TR, so per time, one phase, it's one angle. Per brain region, per TR, then what we can do is compute the cosinus of the differences of these phases. And instead of having now one window, one matrix per time window, now we will have one matrix per TR, so per time in your time series. Okay? And instead of doing these time windows as inputs of the K-means algorithm, we can put these matrices as inputs of the K-means algorithm and try to cluster these matrices that are phase coherence matrices, not time correlations. And what happens if you play a movie of that? Again, here I'm computing this cosinus, the differences of coherence at each TR. We start to look patterns again, as in the previous case, that are not infinite amount of patterns that we see here. So again, it's a good idea try to cluster them in considering these patterns, considering the existence of these patterns. And what happens, and again, this is not a new idea, it's not an idea that happens to me, it's an article that was published, it's a Science Advances 2019, the approach of Atena de Mercy and Suchi and others, that is doing the approach that I present previously, K-means on the basis of phase coherences for patients in coma. Patients in the unresponsive wakeful state, so coma, minimally conscious state, so coma, but from time to time they respond to some basic commands, or healthy controls. And we see that there are patterns that are much more present in the healthy controls than in the minimally conscious state and unresponsive wakeful state. And there are other patterns that are much more present in the unresponsive wakeful state and much less present in the healthy control. What happens when you compare this, when you do the structure function correlation? Exactly the same pattern. Healthy control characterized by a flat line, minimally conscious state, and responsive wakeful state. So the more unconscious you are in a way, the more your correlations or coherences are driven by the anatomical connectivity. Again, it's a very solid result because it can be found not just using one technique, but using different techniques, and not just in general anesthesia, not just in central thalamus stimulation, not just in the macaque, also in humans, also in different states of unconsciousness. So we try to do the same, but using now this approach of coherences to avoid the problem of time windows, and we find exactly the same pattern for the macaques and the propofol. We even recover this reduced repertoire of brain dynamics. So you lose these two states, not by coincidence, but those that you lose are the less similar in the states of unconsciousness driven by propofol. Those two that you lose are exactly the less similar to the connectome. And again, we found again that the state that you lose the most in the unconscious states is the number 7. Yeah, please.
Which measure is this one? Which measure are you using here? Coherence.
This is coherence. Yes.
Coherence. Okay.
Yeah.
So the classic coherence between the both.
No. We start with the Bolt signals. We apply the Hilbert transform. Instead of having, we have now one phase per brain region per TR, And then you apply the cosinus of the differences of the phases.
Okay, yeah, so it's the same as before.
It's the same as before.
That's on the other data set.
Yeah.
Oh, okay.
It's the same as before. Yeah. And again, what happens in the macaque central thalamus stimulation? Flat, more or less, high, and flat again. What happens if you stimulate the ventrolateral? Flat, high, and high. I don't come back. What happens in humans under propofol? Humans now under propofol. Same technique. These are humans in the awake state, same humans in the awake state. They go to general anesthesia and then they recover. Okay, again, this is phase coherences. What happens? Flat, high, and then flat again. And look at the histograms are really similar. In the awake, this is deep propofol. I start to gain this high slope, and then it's more or less flat again. Very similar awake and recovery. Well, and this result can be found also in different, if you use different parcellations like the Shepherd 200. I'm going fast here. So humans in sleep, we can find that you can define more precisely what are different steps or the different levels of sleep using EEG. So this is EEG fMRI data to classify the states. This is a work I'm doing with a master student. And then again, here is less clear, but again, you gain a lot of slope, if you want, if you compare the awake state with N3 being deep sleep. And same story. So it's a very robust of the level of consciousness, this kind of high slope. So second approach, multivariate Ornstein-Uhlenbeck, someone that you know, that has been here a couple of weeks ago, Mathieu Gilson and Enzo Tagliasucci. It's the sleep database that I presented before, EEG, fMRI simultaneously. This is again not a new idea. This is an idea that has been in the field more or less since a couple of years ago in cerebral cortex. This temporal irreversibility as a neural dynamic, as a signature of the levels of consciousness. The approaches that have been using has been mainly data-driven, deep learning, this type of things. Non-equilibrium brain dynamics as a signature of consciousness, more or less the same people, but here is data from macaques in general, general anesthesia. So the idea of non-equilibrium brain dynamics of time irreversibility to assess different levels of consciousness has been around. So, and Mathieu Gilson has this developed this technique to model brain dynamics, model ball signals as multivariant Ornstein-Uhlenbeck processes. So what is this about is about computing some statistical observables from the data and then try to reproduce them using simulations. So trying to fit parameters of the multivariate Ornstein-Uhlenbeck capable of reproducing simultaneous correlations and one-time shift correlations of two different brain regions. And to do that, we use the structure. So we are only fitting the parameters B of the drift of the stochastic differential equations that are present in the structure or in this topological mass. So we can divide, Mathieu developed a method based on gradient descent to fit this B matrix, this D matrix that characterize this multivariate Ornstein-Uhlenbeck process. From this B and the correlations, you can compute a matrix that is called Q, and it turns out that you can compute explicitly, and this is a difference with the previous approaches that are mainly based on data-driven characterization. We can compute explicitly the entropy production of a stochastic multivariate constant dynamic process that can be fit from data. So at the end of the day, we have one matrix B and one matrix B per patient per scan. Okay, so we have one entropy production again per patient per scan. And if we compare the entropy production that we obtain from patients in the awake state and we compare with N3, we find statistically significant differences. And this is not just, we can compute nodal irreversibility, so one per brain region. We can plot them on the brain and we see that it's a pretty prevalent effect when you compare awake with deep sleep. So this is an article that we published a couple of months ago. And the third and last thing that I'm going to talk about today is connectome harmonics. This is a technique also to reveal the structure-function correlation of structure-function interdependence as the state of consciousness change. We know that the brain is driven by different oscillations, and these different oscillations can characterize or change in the different states of consciousness. Here is sleep, but you have more or less the same for coma and for general anesthesia. And in general, low consciousness is characterized by these slow waves in the LFTs. And we have this second fact that the brain show resting state correlations. So this is a problem that is not new at all in the community of physics, and it's usually characterized by this phrase, can we hear the shape of a drum? The mathematics that we use is using the eigenvalues of the Laplacian operator. And these eigenvalues of the Laplacian operators are usually the harmonics. So we know more or less the theory. You are probably familiar with this theory of music theory, first harmonics, second harmonics. We can do more or less the same, but in space now, in the space of the brain. So probably you have seen this type of picture that is how the structure relate to the structure and can form patterns. The mathematics behind this interdependence can be characterized using the Laplacians. So what is the structure in our case is the connectome. We can build a Laplacian from the connectome, and from this connectome we will try to generate the function, but just from the structure. So again, this is not a new idea. This has been around in different papers of human connectome-specific harmonic wave, connectome harmonic decomposition under LSD, so psychedelic drugs, distributed harmonic patterns. This is an article again in the different states of consciousness and how the structure-function interdependence can be revealed using this technique. So what is this technique about? Is taking the connectome, just the connectome that I presented before, compute the Laplacian. What's the Laplacian is the degree matrix, so the weights of the matrix arranged in a diagonal matrix minus this connectome, can be normalized. And then you apply the spectral theorem, so the decomposition of this Laplacian as a multiplication of this orthogonal matrix composed by the eigenvectors of the Laplacian. These are the eigenvalues. This is a symmetric matrix, so these eigenvalues are real. And the orthogonal of this matrix. And with this decomposition, you can compute the harmonics. This is the first harmonic in the human brain, this is the second one, third one, so on and so on. And then what you can do is you take the activity in T1 and you express this activity that is function in a way as a linear combination of structures. Remember that these are the eigenvectors of the Laplacian of the connectome. So what we have here is function anatomy interdependence, so structure function interdependence. So the mathematics behind is well developed. It's analogous to the Fourier transform. We can develop that. and for the connectum and for how it looks like if I compute the energy. So the absolute value square of this decomposition in awake state, the off, so general anesthesia on 3B, on 5B, and control, it's like general anesthesia again. So this correlates very well. These behavioral scores correlate very well with what I found using the connectum harmonic decomposition. And let me repeat again, technique that allows you to assess structure function interdependence in the brain in different states of consciousness. So last two slides about things that I'm not going to talk about today, but I've been also working with. It's multivariate information theory, this whole idea that not all the statistical interdependence, it's encoded in pairwise correlations. You have also triplets, 3 parts of the brain that interact at the same time, and 4. And the tools and ideas to develop that can be found in the mathematics of the multivariate information theory, and we have applied this to databases in psilocybin, LSD, ketamine, and we find very interesting things using in humans in psychedelic drugs with Ruben Herzog, that is now at ICM, Institut Cerveau et Moléculaire at Paris. Ideas in high order interdependence using topological data analysis. This is a collaboration with Fernando Nogregas and the Institute of Advanced Studies in Amsterdam. Again, you can rediscover or develop all the stories of network theory, but for hyper-network theory. So networks are usually pairwise correlations, but there are other structures that are called hypergraphs, and you can develop the mathematics and see differences in the different states of consciousness using this technique. This is a nice thing. Another thing that I not mentioned at all is whole brain models. So how to model using mean field approach the activity of the brain from the connectome and assumptions on the dynamics of the nodes in different species. And again, we are finding very nice results on the different states of consciousness, modeling the brain using either the virtual brain or the dynamic mean field model of Gustavo. So thank you very much for your attention. And this is what I wanted to tell today. Thank you.